Great, wonderful. Yep, and obviously the recording is for other applicants for this past grant who maybe don't have the time to attend today. So I guess I'd just like to begin by saying thank you for joining us today this morning to talk about the exciting new core grant program that Parse Bioscience is launching and Decode is supporting in Australia and New Zealand. Before we begin, I'd like to introduce our channel manager, Sid, just to say a few words and perhaps get us started with the information session.
Thanks, Ibru. Hi, good morning, everyone. My name is Sid Singh. I am the channel partner manager with Parse at the Singapore office. Essentially, I work with various partners across the Asia Pacific and beyond, enabling Parse for the research community. I'm delighted to be here and talk about the core grant program that we are running specifically for Australia and So essentially, we would like to bring awareness about Parse single-cell transcriptomic technology, and that's by the way of demonstration. We would like to enable eligible researcher do their analysis through Parse and generate some good high quality data that could probably work towards either a grant, a bigger grant, or publication. Now, what's on the offer? So, So what we are offering with this grant program is a Evercode WT kit. This kit can actually generate up to 100,000 cells. You may choose to either do nuclei or cells. So depending on your research, you may have a fixation requirement for cells or nuclei. So we would provide you reagents for that as well. Along with these kits, we would also provide you with the data analysis software. You would be trained on the fixation and Evercode as well as the data analysis. So essentially, there would be a lot of handholding through the process where Decode and Parse FASs as well as bioinformatician would help you conduct all of your experiment. And more importantly, we would be working with SAGC, South Australia Genomics Center. That would be doing your barcoding, library preparation, and sequencing. So that's for the overall grant program we have for the winner. We would also be having 5 runners-up. So these would be notified once we have gone through all the applications. I guess with that, I would pass it back to Ibru to talk more about the grant program in details.
Absolutely. Thanks, Sid. So I'm Ebru. I'm the Parse product manager for Decode Science. And Decode Science is the Australian distributor for Parse throughout Australia and New Zealand. And we support a lot of our customers locally, particularly in the last year, to do more Parse experiments and also to support our certified, Parse certified core, the South Australian Genomics Centre, to receive more jobs there. So we're pretty excited to offer this grant now in this region. Some of you may have noticed that Parse have run a bunch of grant schemes, but these have been for overseas applicants. So I'm quite excited to offer this regionally, particularly as our interest in Parse is growing and researchers are planning larger and larger experiments, because that essentially is, one of our value propositions is that we're all built for scale. Essentially. So with this WT kit, you get 100,000 single cells, as Sid mentioned, to be profiled. And we will train you with our local Parse trained FASs in field, which may include a pre-planning call, full training on the fixation kit, etc. So essentially, let's just go for the winner of the grant. Actually, I'll start there. There'll be one major winner that'll win all of those items, as it mentioned, the 100 kit, 1 cell or nuclei nuclei fixation kit for up to 12 samples, and all of the training included for the data analysis. Decoder covering the cost of sequencing, which is fantastic because this is also, an associated cost. And the SAGC is kindly covering the cost of performing the workflow for you. And the 5 runners-up in the grant scheme will also be assessed and notified, and they'll be offered some discounted reagents. So in terms of the logistics, we will train you via video call, etc., and you'll receive, or the researcher will receive the sample fixation kits, and that's essentially to fix your viable single cells or nuclei directly into their fixation reagent. And then we will organise logistics with SAGC of shipping your samples to them to perform. And they will perform as our core lab, and they'll go through the barcoding, and library prep and sequencing, and also some of the standard data analysis to get you to sort of your preliminary data. And then, of course, later on, as I mentioned, we will definitely support you to do more advanced analysis, either using your existing single cell workflows, bioinformatic workflows, or with Trailmaker, our Parse specific software that will actually enable you to do a lot of analysis online without any prior knowledge or prior coding knowledge. So that's fantastic. A little bit more. This is the grant page. It is actually live now. And the beautiful thing about this application is it's simple. It's only 300 words, and we just ask you to be as descriptive as possible in your project abstract. The link is there through the Decode Science webpage, and the submission is essentially just a form, as you can see screenshotted here, where you provide all your basic information, the title of your project, and your detailed abstract. For the first time in our region, we're actually assessing these applications here in Australia with our field application, our Parse field application scientist team, because we now have 2 of them who are on the call, Geode and Scott, who have been fabulous at supporting our customers in the field. So this is some things to consider when you prepare your abstract, obviously as descriptive as possible. So we want to know what sort of sample types you want to assay. Parse kits are verified for human and mouse samples primarily, but if you want to do something a little bit creative or innovative, then feel free to go ahead and submit an abstract about that. And also feel free to email any one of us to ask us questions about that before you submit. And after you submit, there'll be a process of about 2 weeks where all of the applications will be assessed by Geode or Scott for readiness of the project, and I'll go into that soon about what readiness exactly means, the fit of the experiment to the technology overall, so making sure that it's feasible with the technology, and your willingness to do a little bit of co-marketing with us, to present your results to an audience, perhaps in a seminar or a webinar, and sort of to be recognised publicly for this work. We've had some nice examples of showcasing some Australian researchers in the last couple of years from WeHi and Doe that have actually published with Parse and generated some really nice data on the Parse website as part of their showcase program. So it's not only is it a great opportunity to generate novel, exciting data that might push you forward in your grant schemes or whatever, or just to create data for publication, but you can also promote your work by working with us to co-market and to highlight your fantastic research, which is great. So I do believe the abstract page is open for submissions now, even though the official opening date is Feb 2nd, although Hashida might correct me on that. And you have about 2 weeks to submit your abstract. So I think that's a good amount of time for a small 300-word abstract. And on March 6th, the winner will be chosen. And of course, the 5 runners-up will be notified shortly after as well. So just some deadlines to keep in mind. Now, I guess speaking more to the point of project readiness, we're really keen for researchers that want to use this grant to be ready within the first half of this year. So once the winner has been announced, that winner will be placed as free of charge order on Parse by about March 25th. But yeah, they must be placed by that date, and then the kits will be delivered to the customer. As in the fixation kits will be delivered to you, and the library prep kits will be delivered to the SAGC to perform the experiment by June 10th at the latest. So it's definitely a promotion to be exploited at the beginning of the first half of this year, as we're keen for researchers to get going. And to promote the idea of just of pushing forward with some of that research that perhaps have been holding off for because of various reasons, whether it be grant availability or just not being ready. We're keen to support you to push forward. That's essentially the main items and terms. I guess if anyone would like to ask any questions at this point, please do. Unmute or in the chat, whatever you prefer.
Hi, can you hear me?
Hi, Augustine. How are you?
Hi, how are you? Thanks for the presentation.
Yeah, that's
Can
okay.
you remind me how many samples you can fit with this kit?
Yeah, so that's the wonderful thing about Parse. It's quite flexible. So for this promotion, we're only covering up to 12 samples, but for this, yeah. So 12 samples across 100,000 single cells. But usually it can go up to 48. And if that's something that you're interested in doing, then you can just email me later and we can talk a little bit more about it.
Thank you.
Yeah, great question, Hussein. So had a question in the chat. Wanted to double check, are New Zealand PIs eligible? Yes, they are. Actually, we have quite a number of New Zealand customers now using SAGC, which is wonderful for me to see. And the second question, is it possible to do both human and mouse samples on the same kit? I think we don't really encourage people to mix sample types sort of on the same kit. And maybe, Jyoti or Scott, if you want to speak to that a little bit more, you're welcome to.
Thanks, Ibru. So just wanted to the same thing what Ibru mentioned, that we don't encourage. The reason is that Parse technology is based on split pool barcoding technology. So you end up pooling all the samples. So when the library preparation happen, it happens for set of all samples. So the cDNA amplification and the final library, those PCR cycle can only be established using the pool set. So if you have two different samples, one with low RNA content and one with very high RNA content, those things, it will be very difficult to create balanced library and representation of those different sample types. So that's the reason why we want you to use like a single project.
Great, thanks for that explanation, Judy. Much appreciated. Love it. Got some great questions. Are your kits suitable for pig species? Great question. I'm glad you're asking this question because we've had a lot of conversations with various ag research groups in field. Officially, no. Is that right, Sid? However,
it's like we have very first publication using plant samples. So the reason is the cell wall. I think Sid can explain.
Just to answer these two questions, the first one is about the pig species. So if you are doing a whole transcriptome profiling, yes, you can do that. We would need to understand a bit about what's your sample type. So that's where pig species can be done. For plant samples, yes, as Jyoti mentioned, it's slightly, or rather, I would say it needs slight optimization due to the cell wall. We just had a very nice publication from the US universities. So essentially, plant can be done. Again, it's about what plant species are you doing? Are you using a root, shoot, or some other tissue? So we would need to discuss that. But yes, you could do plant as well.
I'm pretty keen, in market to push this a little bit more in Australia and New Zealand, because I think there's a lot of really great, ag research going on, and I've had a lot of questions around single cell for not only pig and plant, but also in aquaculture applications, etc. So yeah, we're keen to support you even in these sort of experimental approaches. Obviously with this grant scheme, but in addition, just with separate meetings where we can discuss how we can make that, how we can support you to do that. Be first in field, I guess, to do that. Let's go over live cells. So that's great. Hussein, great question. The question is, are kits available for live cells, or live cells have to be fixed? All live cells do have to be fixed as the first step. So you create a good quality, viable single cell or single nuclei suspension, and then pipette into our fixation reagent as a starting point. And the beautiful thing about that is that you can keep it frozen at -80 for up to 6 months. So it's fantastic when you want to batch together samples across a time series or from different sites, etc. Yes. So another question. Their research areas of particular interest to Parse Decode to support. Lovely. I sort of partially answered this question. Love the question. Thank you. Medical ag. Yes, both areas. So we focus a lot with Parse on just the pure medical science aspect or in clinical research and basic research, but we are definitely branching out into the ag market this year, again from those questions that we've had in field, particularly as it's quite an exciting opportunity as well for us to be sort of first in field for some of these different ag types with this particular technology. I've personally been bugging Sid for any progress from the US in terms of white papers, etc. for these protocols. But hey, let's just do it here. So whatever you can think of.
So just to give you an overview of, we have successfully, or rather, I would not say it's because it's under publication. We have done multiple plant species that includes maize, soybean, sorghum, Arabidopsis for sure. So these are some of those species which have been done with various tissue types. And also there has been a lot of work in the other livestock, so bovine, fish, insects, all of that has been covered as well. So the beauty is our technology can be applied to any of these species. So yes, we are open to various different areas.
Thanks for sharing that, Sid. That's fantastic. Another great question from Hussein. What sequencing depth are we going to offer? Our standard sequencing depth is about 20,000 reads per cell, which we will offer for this grant. However, you mentioned that sometimes it's competitors' kits, they had a suggestion of 50,000 reads per cell and into the reach saturation. I think, and again, Giordi can speak a little bit more to this, because we have very clear data showing improved capture using Parse technology against our competitors. You don't actually need as much sequencing depth. So 20,000 reads per cell is where we would recommend beginning. However, we can speak to you about your particular research question. Obviously, some people perhaps are looking for rarer transcripts or whatever, or rarer cell types and may need additional sequencing. And we can also support you that way as well. Being MGI sequencing provider in the region, we do partner with SATC to get some really cost-effective sequencing on the end of this grant. So yeah, we're definitely open to exploring deeper sequencing, but 20,000 reads per cell is where we start. Did you want to talk any more about that, Jody, or have I covered most of it?
You already mentioned that depending on the project need, we can discuss that. So it should not inhibit you to submit your project, basically. So that's the short answer.
Yep, great. Another great question from Kenny. Does the kit from the grant allow sequencing of archival FFPE or FF single nuclei? So this is a brand new product that Parse's released at the end of last year. We had some marketing about that in December. We're still sort of waiting for more information about this kit ourselves, and in terms of the full sort of pricing and disseminating a bit more of that information. This kit doesn't include that, right? So this is just a plain, no, this is unfortunately it's a plain WT Decode kit, not the reagents that are compatible with those particular samples. Did you want to say a bit more about that, Sid?
Yeah, that's true. So this grant program is focused more towards the whole transcriptome profiling and not towards the FFPE samples. However, what we do have right now is we are putting in all the researchers on a wait list because we are about to start shipping the FFP kit. So if you are keen, we would definitely like to discuss with you and offer any specific type of discounts possible.
Great, thank you, Sid. Yeah, that's a really exciting new addition, which hopefully will get a lot of people on your wait list. I think we've already had a fair bit of interest in there. So please stay in touch, Kenny. Great. Another question or comment from Hussein. I love how chatty Hussein is. What is the particular criteria you're looking for a winner for? In the winner, for example, does the science need to be novel, exciting? Does the experiment need to be feasible? Is it good if there is pilot data track record? Yeah. Great questions. So feasibility is quite high on the criteria. And then probably the novelty, I guess, of the science as secondary. In terms of, Sid, Jody, in terms of track record of PI, or if there is good pilot data, that's not important at all in our, I love that you're shaking your heads. Yeah, because
Yes.
essentially this is a very sort of bit of an open access scheme. Like we just want this in the hands of researchers to get on with it and do the science that they want to do. Which is why I'm really want to push more of these, private type grants, because it's not the same game as grants that are applied through traditional sort of public funded avenues. We're a lot more flexible and we're willing to work with you. So yeah, definitely feasibility and yeah, a bit of novelty as well.
Yeah, I would like to add one more thing, that readiness of your sample. So in the project, you should be able to start sooner than later.
Yeah.
So we don't want you to you won't have sample ready. So readiness of sample is one of the another criteria besides the feasibility. It's a part of feasibility, but that is one of the another parameter we'll check, whether you are ready to do this project now or maybe 6 months or a year later. So those people will be given priority.
Yeah, great. Thank you, Judy. And of course, about that, this is just the beginning of the discussion. If you do have projects that you are planning 6 months or further into the future, please keep talking with us. That's essentially what we do in field a lot of the time, is do a lot of forward planning for researchers. So yeah, we can have both discussions, but for this particular grant, yes, readiness is very important. It is good to be chatty and say. It's very good to be chatty. Fabulous. Love it. Any other questions this time? For all the questions, I'm glad everyone asked them, because those were sort of the main ones that I was expecting. Yeah, and of course, we're open to more questions and queries over the next 2 weeks as the grant period is open. So don't hesitate to get in contact with us. Oh, great. Thank you, Hashida. Oh, no. Do you have support personnel in New Zealand? Hussein's still chatting. Yes, we do. So Auckland Genomics is a site, Jody, that you train personally, right? And I believe Nikki Free from there has been accepting quite a few, not quite a few, but a few more past projects this year. We've really wanted to support her to get tracking and build up her clients, her past clients. So although this grant is directed to SAGC in Australia, it's because it's our one, major certified passport. We definitely have an interest to build up Auckland Genomics and the support there. And we are actually actively doing that, right, Jody, with a few.
Yes. Yes, so we are actively working with Auckland Genomics, and they have completed their own projects like last year, and with some good data. Now they are ready to take as many projects. So I think this year we'll be seeing more projects coming, service project coming in their way. And
yeah, it'd be really nice to get them ramped up as well in terms of incoming projects. It's our main aim. Thank you, Hashida, for sharing the get notified on past grant open date fill out form link in the chat. That's great. because, you know, we're always flooded with emails and notifications. But yeah, it'd be good if you get any calls about that. Oh, great. That's wonderful. Lovely to hear that nice feedback from Nikki through the same at Open Genomics. That's awesome. Great. So we're coming up to our half an hour time point. I'll just leave it for maybe another couple of minutes for any other further questions.
I think if there are no more questions, maybe we can call it a day.
Absolutely. I think everyone's had their say. Please apply, get creative, and of course, you know, reach out to us with any further questions that you have. We're here to support you to get moving with your Parse experiments. I do also realize, like, it's lovely to see, you know, some customers here that maybe we've had contact with previously, but there are a lot of sort of new names, new faces. So if, you know, if you would even like just a general introduction, and perhaps you haven't had a proper introduction into the technology or the Parse tech, we're happy to jump on board with you before or over the next couple of weeks to even just do a general intro to be prepared as well. So great. Thank you everyone for your time. I believe Hashida will probably post or circulate another emailer with the recording in the coming weeks as well for you to watch again and to share with your colleagues. So please do. And we look forward to reading the grant applications. Thanks so much.
Thank you for your time.
Thank you.